Hello, everyone. Welcome. I'm Jacqueline Fabius, the Chief Operating Officer for the Quantitative Biosciences Institute, QBI, and I'd like to give you a warm welcome to today's event. Today we're kicking off our third joint event with the Scientific Department of the Embassy of France, and specifically the French Consulate of San Francisco, on the topic of research and health. Our first joint event was in 2020. It was virtual due to the pandemic. And I like to say that we were slightly ahead of our time, with the topic being AI, big data, and health. In 2021, it was still virtual. It was a multi-day event titled Reshaping Science and Health. Each year has been a great success, but I'm happy to welcome you to our first in-person event today in 2023, HealthAI. And with this, I'll now pass the mic to our partner, the French Consulate of San Francisco, represented by Emmanuel Pauliac-Vaujour, the Attaché for Science and Technology.
Good afternoon, everyone. I'm Emmanuel Pauliac-Vaujour. I'm the Attaché for Science and Technology at the Consulate General of France in San Francisco. It's my great pleasure today to be your co-host on France's behalf. I would like to thank our longtime partner, QBI, and Jacqueline for hosting us and co-organizing this event with us, which is a French-American event, so you will see a lot of French or French-speaking people around, and that's normal. I don't think I need to convince you that the topic of today's symposium is of utmost importance. We're proud that France can contribute to these advances hand in hand with a renowned partner like QBI. I'll try to keep the speech as short as possible, but I have a few things to say. One of our main missions at the French Office for Science and Technology of the Embassy of France, along with all the scientific attachés in Atlanta, Boston, Houston, Chicago, Los Angeles, and Washington. Our objective is to foster scientific collaboration between French and US researchers and support students and research mobility between our two countries, especially at the doctoral level or early stage of the career. It's not just by chance that the collaboration between France and the US is one of the strongest and the most enduring. Our two countries do have a lot to share in terms of knowledge and know-how in all aspects of society, including science and research. We also value the supportive entrepreneurial spirit that exists in the tech world and which is particularly strong in the Silicon Valley, as some of you may know. So incubators, startups, spin-off companies, and large industries are the ones that truly turn the most mind-blowing research into a reality that will impact and improve the lives of the greatest number. We could not be more thankful to see French and American actors of innovation come together in an event like this one. And none of this would be achievable without their kind participation. So I really want to thank them truthfully. My final message for today is that this symposium is a good example of what we can achieve with all of your support, as well as with the French and the Francophile community. So it's only one example among many initiatives supported by the French Consulate. On an everyday basis, our goal, our job, is to listen to talk about the scientific and technological subjects which are of crucial importance to you and to both our countries. And the future of healthcare in the AI era is definitely one of them. We aim to create dedicated spaces and communities where scientists can meet, share major scientific results and insights that will impact the research, the economy, and the society significantly. There are many ways in which you can support our actions, whether you are French or not. So please come and talk to us during these 2 days, learn more about the things that we do and the many ways that you can get involved. And just a quick word to maybe mention the photo expo that you may have seen when you came in this room, which was one of the initiatives of the Office for Science and Technology last summer. And we actually had some laureates who won some prizes in this photo competition. So next year, please participate. Without further ado, I leave the floor to Tanya Cortem, who will also introduce our first panel discussion. And I wish you all a very fruitful and enjoyable symposium. Thank you. Yeah, good afternoon, and welcome from me as well. I am Tanya Korteme. I am a professor here at UCSF in Bioengineering and Therapeutic Sciences. I'm also the Vice Dean of Research for the School of Pharmacy, and personally extremely excited about the confluence of AI and discovery science, reaching all the way from really fundamental curiosity to impacts on our everyday life. And so to me, some of the most exciting advances happen at the intersection of science and technology, of the intersection between different disciplines in science, and at the intersection between technology, science, and healthcare. And so I think that's why, to me, what we're witnessing all over the world, but also what we've been discussing in the last day and a half in a sister symposium on AI across biological scales, and what we are continuing to discuss today in the panel discussions, will be really at this exciting confluence of advances in computer science, in cell biology, in drug discovery, in healthcare, and in clinical applications. And so I'm very excited to hear from experts in this area how they are viewing all of these developments and what they can inform us on what the future holds in these areas. And so with this, I'm going to introduce our first panel moderator, Lisiena Hysenaj, to introduce her panel on cell biology and AI, and also the panelists. Welcome.
Thank you very much. Hello. So the first panel will be AI in cell biology, and we'll have 4 speakers. The first one is Michael Keiser from UCSF. He's an assistant professor at UCSF. I think this microphone is not working. Okay. Yeah, okay. And the second speaker is Laurence from Calzone. She's coming from France, from Institut Curie. And then we have Ossine Lourdani, that is an engineer on AI, and Audrey Smith from ML Twist. So every one of the speakers will present itself for 3 minutes, and then we'll have a panel discussion together. So maybe, Michael, you can start.
All right. Maybe we'll put this. All right. Well, thank you. So I'm an associate professor here, and my background is originally in computer science, and over years I actually came into this area through UCSF itself. I was once upon a time a bioinformatics graduate student here. And so the lab, and we're giving very brief intros, our lab focuses on 3 major application domains in biomedicine and biological research. For us, AI and ML questions are at the center of it. And so we look at molecules, and we spoke a little bit about that yesterday, about perturbations. And here, when I think of a perturbation, it could be a molecule, it could be an environmental factor, it could be a functional genomic change to a biological system. And so some of the areas we've been interested in have been cell biology, as relates to this panel, also whole organisms and collaborations in zebrafish and looking at neuroactive activity. And then imaging. Imaging can span so many things in our area of interest. Here I'm showing some examples of collaborations in the space of digital pathology, where we work on becoming a force multiplier or a way to help pathologists operate at a scale, resolution, and generalizability that otherwise would be impossible to do as a human. And so we were asked to show 1 or 2 examples. I'm going to take the easy route and show 2 that were from the presentation I gave yesterday. The first one is a generative AI model where we were ingesting images, these are microscopy images from cellular screens for a neurodegenerative disease phenotype specifically the tau protein. And at the left, what you have are two different channels of the same field of view of these cells. At the top, you see YFP-fused tau protein. And at the bottom, we have some nuclei. Now, as I mentioned at the time, one of the big problems with live cell imaging to do drug discovery and other screens at scale of hundreds of thousands or more perturbations is we can't put antibodies in that system. We can't run it in a fixed cell format. It doesn't scale. And so if you're doing this in live cell, and we also look at different time points, we're stuck with only seeing all of the tau. What we really want to see, though, is the misfolded tau, or the tangled tau. This is the tau that builds up pathogenically, and we see it in the brains of patients who have passed away from Alzheimer's disease. And that's only some of it. And so what we were able to do was train a deep learning method that could take data from screens that had been done years before, and ingesting those two input images on the left, generate what we wished we would have had all those years ago, and specifically focus on the tangled tau. Happy to talk about that. And then the other sort of result that illustrates area of interest for us is a second generative approach. And so this is a generative diffusion model, and what we're able to do is generate fragment by fragment, piece by piece, molecules within protein pockets. And so combining these two approaches, you can imagine a whole pipeline of testing, experimentation, and assessment in order to move forward understanding cellular diseases and drug discovery. Thank you.
Thank you very much. And so now we are still in academia, and we'll have someone that comes from France. Laurence will introduce what she's doing and who is she.
Thank you. So my slides are not as beautiful as what I hoped, so my name is Laurence Calzone. I'm a researcher in Curie Institut. I coordinate together with Emmanuel Barriot, who is in the audience, the group of computational systems biology of cancer. I'm a mathematical modeler, and with the focus on cancer applications. And what do I do? What am I interested in is to build mechanistic model that describe the biology of the disease, and in our case in particular cancer, but not only, with the purpose to understand the impact of the alteration, suggest optimal drugs, and predict drug response or drug synergies in a patient-specific manner. So where do I come from? So I'm a mathematician by training. I started doing chemical kinetics in particular, and then I moved to biology through theory, and I did a PhD in theoretical biology here in Virginia Tech, not here, but in the US. So my first interests were to do some dynamical modeling of the cellular processes, and then when I moved to Curie, the type of data we were dealing with could not be applied to ordinary differential equations, really. So this is when we developed some other formalism based on Boolean approach. But what we really develop in the group are models of tumor evolution and expansion, taking into account space and time, so with a big data and a lot of parameters that we don't have. We also develop some tools to model individual cell populations, and we want to integrate, of course, all the omics data that we can into our models. So I'm really interested in mechanistic models and how to make the link with artificial intelligence models. You know, we try to explain really the type of results we get with AI models. And we can do that from two perspectives. First, as an input to the models. So we learn from the data to build our mechanistic models, of course, with the stratification of patients, but also help us infer the models that we build, both the networks and the mathematical equations from this data. We want to integrate multimodal data as model parameters. So again, learn the parameters from this data, and extract features from the data, which pathways or which genes really separate the response to a treatment or not. And then when we have this model, we are also interested in AI to improve the models. And how do we do it? We search for optimal parameter sets. So we have a lot of parameters that we need to infer, and this is really difficult to do it by hand. And of course, we expect to use AI models like surrogate models to mimic the complex and the computational heavy models that we build. And we also use AI to generate synthetic data for model optimization. Sometimes we don't have enough data, and this is one way to augment the data. So we want to make the link between AI results and model outputs and really fill the gap between mechanistic models and AI. And I think I will try to give this perspective in the response to the questions that we will talk about. The point is to explain the content of the black box as much as possible. And I was also asked to give two projects on which we work in the lab. The first one is called TPIT. The point is, it's the holy grail to try to find some biomarkers for response prediction to some treatments. And here it's about immunotherapy. And we use multiple data types to increase the power to be predictive and explain as much as possible why these patients respond or not. And this, of course, opened the road to spatial modeling, which brings me to the second project, which is my expertise, is spatial modeling of tumor invasion. We model both intra and extracellular interactions to simulate in silico treatments. So we start from data, we infer the model, we infer the parameters, and you can see here an example of a tumor and the invasive that starts invading the tissue. And then we can play with these models and search for optimal drugs in silico that we can then test in experiments. And I will finish here.
Okay, thank you. Now it's Austin. It's a PharmD that is gonna explain us more what he has worked for some companies in the Silicon Valley on AI. So he will introduce himself.
Sure. Hi everyone. Thank you for inviting me, and thank you for being in the room today. Really appreciate it. Quick introduction about myself. My name is Hossein Lordani. I am a PharmD, indeed, by trade. I am originally from France, which I wouldn't mention usually, but feels relevant today. Started my career in France, actually, in pharma and biotech, and went to business school, moved to the Bay Area a bit more than 10 years ago. And since then, mostly spent my time building AI products. And so I'll try to bring that perspective of a practitioner today and somebody who has led the development and the commercialization of AI products in different industries, in particular healthcare and life sciences industries. So a couple of examples I extracted here is the work I've conducted with the teams at Autoris and RapidAI, where the goal was really to leverage AI in a clinical setting to build software medical devices to support clinicians in their diagnosis and treatment decisions. So a very specific set of challenges are related to that. It's really about the measurement of certain clinical factors, the detection of certain pathologies, and that's a very specific type of challenges that I'm sure will come up in our discussion. Most recently, I've worked at DeepCell, where we took a very different approach. And DeepCell is a company focused on research, especially at the single cell research, and we applied AI to study the morphology of cells. So we built an end-to-end platform able to take images of cells in flow and extract high-dimensional information from these images and help researchers characterize and make sorting decisions using the outputs of our foundation model. So a very different set of challenges here. And so I extracted just a few thoughts that may color my intervention in this panel, very high level, and I won't take too much time. One is I'm a product person, so I'm really focusing on building products and solving concrete problems. As we know these days, there's a lot of hype, a lot of excitement, and for good reason, around AI. But beyond buzzwords, beyond the hype, I tend to focus on identifying very specific concrete problems and focus on the impact that AI can have on solving that problem, with an emphasis on the interpretability and the usability of these models. Because the best model in the world is not very useful if the end user cannot really use it in good conditions. The second element I kind of touched on is that challenges in AI product development and commercialization implementation are very different depending on the industry. In particular, as I mentioned, clinical environments versus research-only, research-focused environments present very different types of challenges, and that may color my intervention today. And finally, one point that I wanted to insist on, and actually that resonates a little bit with what Laurence, I believe, was presenting, is that one thing I've observed recently in this and in my time working with biologists and researchers is that there's a persistent cultural difference and difference in approach between researchers in biology and AI and the AI community at large, and it focuses a lot, in my experience, in explainability and in whether or not a model actually describes a mechanism. And of course, this is a complex topic, so I won't, this is way too high level, but I want to emphasize here that sometimes that question is very important, and of course, a model should be explainable, and at the same time, the value of a model can be in its predictive power alone. That happens, and an example that I didn't work on personally, but I think many of you in the room can relate to or know of is AlphaFold, for instance, this model trained by Google DeepMind to predict the 3D folding structure of a protein based on its sequence of amino acids. There is work ongoing on explaining exactly how the model works and how it's making its predictions, but the value of the model in unlocking avenues for research just by being that potent and that efficient in predicting the structure is already huge, and sometimes explainability is not the only aspect that we should focus on. So I'll stop here. I took already too much time. Thank you, everyone, and I look forward to the discussion.
Thank you, Austin. So now we continue with other 4 panel, and she's Audrey. She's a woman founder, and she has built this company that is on AI services. She will explain more about it.
Hi everyone, thank you for having me. So 2 facts about me: I'm French, as you can probably hear. And the second one, I'm the only non-scientist person in this panel, so very happy to be here, very excited to be here, but I might offer another point of view. I founded MLTwist with my co-founder 3 years ago, and the idea is really to help solve a very boring problem, actually, but that is like very persistent when you start training a model, is on the fact that there are still like manual tasks that still need to happen when you need to create data processing or data augmentation, and when you try to not only build data pipelines for a certain model, but also maintain it because it can break over and over again. And that type of work actually is taking 80% of the time of any data scientist, which is a lot. And most of the time, if you talk with data scientists, what they want to do is spend their time training a model, not doing that janitorial work that we're happy to take over. If you look about like the yellow chevron, you're going to see that as of right now, a lot of technologies happen. A lot of technologies have like raised over the past decade. But what really is not really like you cannot see happening is that you need to build a data pipeline between all those yellow chevrons to make to train your model, and that's something that still not have been like really solved as of today, and that's what we focus on. If you look at the work, the manual work, if you have a technical person on hand in your company or in your work in academia, you're going to have to go through all those different steps to get your data ready for a machine learning model, to ingest the data and get trained on the data. That can take up like several weeks, as you can see. And we build a platform that can auto-generate data pipeline, and all the purple steps that you can see are done automatically by our platform, which help people like you or data scientists to just focus on, like the exciting stuff, which is working on the accuracy of the data or just training your model and checking the performance of the model. As you can see, we have been lucky enough to partner and have also great customers that we are allowed to talk about. So we are working with Sandia National Laboratory, but also UC Davis Health. We are also working with Stanford HAI department and partnering with Capgemini and other names on this slide. And so we have been able to work on a lot of different data types from what Osin was talking about, like images, but also videos. It can be also text, and it can be also DICOM. We have actually worked on DICOS, which is a spin-off of DICOM. And that's about it. That's us in a nutshell. Oh, you can see like a bit of our accomplishments too. But yeah, that's about it.
Thank you.
Thank you.
So, okay. Thank you very much, everyone. So as we saw in this panel, we have people from academia that work really on building this model and making sure that we'll have some discoveries from these models. And then people from industry that are making sure to create a product that can be used for everyone. So now the question I have, it's what is one of the most impressive discoveries that we have had in the field of AI and cell biology in the recent years that we could not have had just with humans alone? Anyone wants to answer this question?
Maybe. Can you hear me? Yes. Maybe I'll get started just because I just reused the example I took actually. I mean, in my presentation. I think AlphaFold is one of the most impressive developments that we've seen lately. It's interesting to see that it's a collaboration with academia initially, with EMBL in Europe to build the database, and then the development of the model was conducted by Google DeepMind. To your question, Lisiena, so the problem of protein folding and the 3D structure, the prediction of a 3D structure of a protein is a well-known problem. It's been worked on for decades. It's been worked on tirelessly by a number of groups, and I'm sure many of you are in the room are aware of it, so I won't spend too much time on it. But the level of performance that was reached recently in 2018 with the first version, and then 2020, is just something that we couldn't have reached without the latest development of AI. Now, when we say would not be achievable by humans, I'm always a little bit, not sure what we mean. We always use mathematical modeling. We always use different tools, and these are created by humans, and AI are also created by humans. So I would still, put that on the credit of humanity. But this is, I think, a very solid example.
Okay. Thank you. So another thing, this is like maybe in the mind of everyone, we have seen all these sci-fi movies where we see that, okay, something, it's taken by a malicious attack and some criminals took over our mind, that in this case it will be our machine learning. And I was thinking this when I heard the talk of Michael, where you were talking about deep learning techniques that you're using in the lab and how you can determine, so it's like a tau protein that's misfolded or all this. So how you make sure that someone will hack your, how are we sure that, what do you do to protect your system and imagine someone comes, hacks your system, and then your students or PhD students or postdocs will use a model that is not right and will claim discoveries that are false?
Well, so it's an interesting question too, because in AI, there's so much of a history of releasing our code. This is crucial if you want to publish at a computer science conference. If you want to do any of these things, you have to make what you're doing available. And we've seen questions about this with things like ChatGPT and the question of guardrails or improper use. But when we think about drug discovery, there have been papers where people have asked, what about old techniques? What about things we've known for decades computationally? How hard is it to misuse a model? And that has always been possible. It hasn't been the computation that's kept, the computational barrier usually, that's kept someone from making a very bad drug. It's been the synthesis, the construction, all of the other steps where the rubber meets the road, where there's actually a lot of oversight and a lot of institutional support. And that is one way that the models themselves are less at the center of that question.
But what you are afraid on? It's not the model. It's like this malicious attack don't exist. It's just like we are scared of it.
I am less afraid of models than I would be about how people choose to use things.
Okay.
And so for me, I'd always take that answer to our organization, to the universities, the companies, oversight, accountability. That would be the place that I would ask, because the flip side of this is I remember years and years and years ago, I was a student and someone was presenting on some new technology or result, a professor who was visiting, and it wasn't published yet. They were hoping to submit it to a really high-end journal, and someone said, "But aren't you afraid that by telling us all about how to do this, someone's going to scoop you or use it in some way you don't like?" And he said, "Young man, I would be thrilled if someone would try to steal my work."
Okay. Great. So thank you for your answer. Yeah.
Just one thing about, in academia we share as much as possible, so we put on GitHub everything we can, whenever we can, to make sure that also we get the paternity of what we develop, but also, like you said, we hope that people can use it and then make it better, and then we all work for the same purpose.
Okay, thank you. So one question I'm having now, because I was searching a little bit about ML Twist and your collaborations, and so that I thought ML Twist is a service and it's used only by companies. But I saw on your website that you collaborate with Stanford and Berkeley, and I was pretty surprised about it. Can you explain how you help these big academic institutions that generate a lot of discoveries in AI to improve their data?
Sure. So we have several ways to work with them or to partner with them. One of them is actually using the students at those universities that are medical experts, for instance, and who can help us do some labeling for some models. We have other things, like you said, for Berkeley Labs, it was a bit different because we were working on a grant for the Department of Energy, and we were able to collaborate with them on a plan to win actually the phase 1 of those that sphere. And every time, so we also working with Stanford HI, and that's something else. They are working on creating some models, some LLMs internally, and we are generating the data for them. So that's we are very, very proud of that. But every time we talk with an academia, it's going to be a different need. But we're happy to explore any way we can partner with academia.
Okay, thank you. So basically, we saw that Michael said that everything the discoveries are done and the models are not misused if people use them properly. And then we have some people to help label well the products in order to generate even more discoveries. Now, as a cell biologist or immunologist that I am, the question I am asking is, I've heard all these different models and companies that exist. Is there any way I can learn, because I don't know how to code, I can learn how to use, or where I can be informed about these new technologies that are coming out?
It really, I guess I'm coming at this question from again, product perspective. I tend to believe that not everybody needs to be trained on the technical aspects of AI, and that is the role of companies largely, but also, of course, the contribution of the academic community to come up with tools that are very easy and intuitive to interact with, that in the background do very complex things and use a lot of code and use a lot of very complex mathematical concepts, but from a user perspective, just kind of shield all of that from users. I don't see a very, I may be incorrect and you may have a different opinion, I don't see a very strong argument for AI to be any different than software engineering in general, for instance. We're not asking everybody to be able to code, as you just said, in research, and I don't see why that would be different from AI. I think the argument is a little bit different when you think about students, however. When we think about students in the fields of biology, I think the the intersection of computational biology, AI, and biology in general is reaching such a level that there is probably a better argument there to say that the next generation in general should touch, at least to some degree, AI training. But it's a field that's evolving so fast anyway that it's something that is a very complex topic, and I don't have the expertise on it.
Thank you.
No, I would say that the key for any project is collaboration. So as a cell biologist, you don't need to know AI, but you need to be surrounded by people that can help you and that talk the same language as you.
Yeah, so this is the question. It's like, where do I find, because every time I go AI and then you have this courses, I'm like, oh, I'm lost. What do I do? So the question was like, where can I find this? ChatGPT, for example, was all over and everyone was talking about it. So I started to use it and it was easy and saved my life. But is there any website or something?
Well, I would emphasize the collaborate point because that's what I was thinking too. Because actually, I don't think ChatGPT is the right metaphor for how we often want to use AI for high-stakes problems. Okay. Because instead, I think of someone who uses the centrifuge without training, or the fax machine early on, and now it's gummed up. Either you're working with someone to at least get yourself started, or there's a core facility, or there's a way to get the training.
Okay.
And the problem with AI is less frequently that we would gum up a machine or do something to the rotor. It's more commonly that it would appear to work in a way that was misleading or not useful, because it is a pattern recognition thing. It will always find patterns and exploit them. So collaborate.
Thank you. And so I'm wondering, just like you are all open to have a contact from someone from industry and build a new model or help them on AI. This is great. I think it's a message we have to show. So I think we are, I don't know about the questions, or I have 5 minutes. Okay, perfect. So you'll tell me whenever. Yeah, I have more questions. So another question that I had, it's, okay, we're in the same space, we need to be trained. Do you think like a training of AI, like how to use AI and not to misuse it, should be mandatory in every company now or in every lab, like the safety training?
I'm not sure exactly how it would look like, but I think that it would be very important to start educating people on the good and the bad, starting by data bias, which is like something that is like, as human, we all have biases, but if we are aware of it, if you let people know that they need to pay attention to the data they're going to be selecting for their model, then there might be a chance that we're going to reduce the amount of bias happening in the model. And then to go back to your question where you were talking about like learning how to code on what it means to do AI and so on, I think it's actually important to keep that separate, just because you're going to have people from different backgrounds collaborating, and they will fight also biases that way. If you have everyone, educating the same way, learning the same way, in the end, you're going to have a lot of biases.
Okay, thank you.
Just to say that, in the company you need a wide range of expertise.
Yes.
Again, so you need to have people dedicated to that, and the other ones knowing the limitations of the results and how to use these results is really key. I think this, if we have to, all of us, if we have to learn something, is the limitations and the biases, to be clear about the biases.
Yeah.
And just one thing to say about it, because of the strength of all the different expertises and opinions, we don't all need to be programmers or developers of AI. What will happen more and more is these tools in different ways make their ways into the workplace, into the research environment, all of these. And I worry that it seems like when you read articles, when you think about, you talk to people who have questions outside of the space, it seems as though it's categorical. Do I trust AI or do I not trust AI? Do I use a prediction or not? But when you think about things like ChatGPT, which I'd like you bringing up earlier, we are used to speaking and writing, and we can critique it. We can look at it. We know sometimes when it's wrong, and if you try to look for that, you find it more and more. And so I would say the training that would make the most sense is how to be a critical user of it.
Yeah.
and what techniques to have to test whether this thing is telling me what it so confidently is presenting.
I completely agree with that point. And to take it one step further, I think one thing that we need to put the agenda even more is the question of open source in the industry in particular. Because indeed, in academia, and we mentioned it earlier, there is this culture and this requirement to share your results, to share your code, to share your data. And in the industry, it's not always the case, and for good reasons. Of course, if you invested a lot of money in training systems, you want to protect your IP, you want to protect your first mover advantage in the market, and completely understand that. But we need to find ways to still have enough data available and enough source code available for researchers in academia or not to actually validate these models and test them and be critical about their performance.
Thank you. So basically, I'm more optimistic now on the use of the AI and the models. So now, just to talk about cell biology in general, what is, as an immunologist, I'm more like in characterizing the cell types, and it's basically what you have done a little bit in deep cell. So I'm like, will we be a moment we will just recognize cells by only one feature, just for example, imaging, or we should still do our flow cytometry with 300 different colors and then analyze by ourselves, and how far is this? I've heard also, like in the same space, I've heard also about wet lab. So if you can explain this and where we are in cell biology right now, and what's the...
Yeah, I can draw from my recent experience at Deep Cell to at least answer the first part of your question. So we are at a point right now with the performance of AI models on cell imaging, and we can really extract high-dimensional information in a way that is consistent. An improper way to say that would be to sequence the morphology of cells and at scale, right? And that's completely new, and that really challenges or subverts, let's say, the existing methods in FACS and flow cytometry. Where I think it's very interesting is that currently the way to characterize cell types is based on the taxonomy in biology that is in evolution, but still very much rooted in surface markers, cell markers in general. And getting out of that taxonomy initially is a little bit different when you start looking at phenotype. And that's the next frontier, is really looking at phenotype for itself and really leverage that as a new omic, basically, type of data. And the next frontier after that, I think, is multi-model research, where you integrate that data with multi-omic information from different sources. So that's I think, how you get to a holistic view of a cell that takes very different lenses to analyze it. Did I answer your question?
I think so. Maybe Michael has something to add about.
Well, I very much agree with what you're saying. And what I was also kind of reflecting on and thinking about this is, you asked kind of when we're there. And I remember 1965, a paper on strong inference. A quote from it is, "The measure of a method is its use." And so we're talking about methods and technology and platforms, AI or otherwise, are combined in this hybrid system. For what use? And so for some uses, we might already be there right now. And for some things, we cannot see them by imaging without the right marker.
Mm-hmm.
And we don't know that yet. And so there's this interest in multimodality, how many things we can bring together without breaking the bank on the experiment in the first place.
Yeah.
And so I would actually turn the question back to you and ask, for what use, and what is sufficient? And
I was thinking more on discovery in cell biology. For example, it could be used in a biomarker space. So for example, I would imagine an AI model that could help me realize what cell types I have based on some phenotypes that I see on the microscope, or what can be the molecular pathways that are affected on these phenotypes. And I saw that you are doing something like this maybe in the lab to link this maybe also in pharmacology. And this is how you see this field, and what are the discoveries in there.
So this could be an hour conversation. I'm sure others have thoughts on it too. In short, differences between supervised and unsupervised. We'll go and see what's there based on the sort of information we are already tooled up to collect, say images and 4 channels, compared to I have a really specific need and a priori bringing in my human expertise. I know that none of those image channels are likely to get the job done, and if they did, I'm actually kind of worried. That would actually be a good counterfactual test to make sure I couldn't get that out of an AI model. And in that case, maybe I need transcriptomic information, or maybe I need a different modality. And so I would say that the best way that we can answer that is empirically. And that's what we do as scientists all the time. We design experiments and test them, prototype as quick as possible. This is no exception.
Anyone has to? Okay, I have just one last question. I think when I was talking with Miguel, he talked to me about this red army, I think.
Oh, red teaming.
Red teaming. So how this works?
So this was this question of how do you know whether to trust a thing? How do you know whether something is hallucinating? How do you know whether the prediction is worth looking at and investing what could be months or more resources and time? And so there's an analogy that we can bring to bear without having to invent new ways to think about these questions. We can borrow from cybersecurity. And so in cybersecurity, you never know if your computer is safe. You follow all the best practices. You update the software as best you can. You shut down all kinds of access that you don't want. But there could still be something that you never thought of. And that's one person's computer. What about an organization? What about anything else? And so in the field of cybersecurity, where it is impossible for guidelines or best practices to guarantee safety, in the flip side, in the field of scientific AI, there is no guarantee that a model works unless you've done something to try to break it. In fact, unless you've done a lot of things to try to break it. Unless you've scrambled your data and trained the same model to make sure it doesn't work, because that would actually be bad. It would be cheating in some way if it did. This is the field of shortcut learning, where models learn shortcuts or artifacts or things that we didn't anticipate or that we didn't see. And so it's that concept to be an active adversary to the thing that you want to succeed. And we already do this in the lab, if you think of controls. But now we need stronger controls because it will try to do what we ask. And that's the difference. And so that's what red teaming is in cybersecurity, and that's how it could be applied in scientific AI.
Thank you. And I wanted to ask more for the industry. How industry makes sure that you have a red teaming? Do you have like a team that works on making sure that you're...
So not at Emetris, because we are generating the data, we're not generating the models, but that's definitely something that needs to happen for our customers. So we work with defense, for instance, and that's definitely mandatory to have like some ways of trying to break the model. But you can do it yourself. You can just go on ChatGPT and then ask questions again and again, and you're going to see at one point it's going to break. And you're going to even see some biases appearing like very quickly on what a pilot is. It is a man or is it a woman or a nurse? And you're going to see that this is actually all coming from how the model was trained and what data was used to generate ChatGPT. So that's very important, and that goes back to what you were saying, Michael, that we need to be able to understand that whatever we see that is AI stamped, we need to criticize it. We need to be able to look at it with an eye of, hey, is it real or is it biased, or can I really trust what it does or what it says? And that's like something that should be done even at the school level, like with our young kids, because with like everything that is generated right now, we don't know what is true and we don't know what is not true, and that's very important to train kids and students to keep that eye out for that.
Do you?
No, I agree, but this is a scientific reasoning, right? You want to see how far you can go and find the weaknesses of your models, and this is something we do. Even when we read an article, we don't we don't take it as it is. We want to question it, and I guess this is what we need to keep in mind with AI models. They're not the truth. They get closer to what we want, and they answer a very specific question, not universal questions.
Thank you. So now, as we heard, we should not be scared of the AI because they work in the same way as we work for an experiment. So they check every step and they do a checkpoint. And also, they're all working in a collaborative way, so we should not be afraid to go and ask them if they're in industry or in academia. And then more discoveries are coming as the world progresses. So now, so I think for this nice panel, and now everyone, if you have any questions for the panel, you could, I think we should line up here, right?
There will be a microphone here, so you can line up and ask questions.
I can start. Thank you so much for the panel. So I have a question because I see a lot of like academic or pure tech kind of players. How do you work with the biotech industry? How are your solutions implemented? Do you feel that there's any bottleneck from the pharmaceutical industry to like adopt all these great technologies?
Right. Well, so one of the best answers I can give is the second result I showed with the small molecules is actually a joint development with Genentech. And so Genentech partners came to us and we worked on a close collaboration for more than a year where there's both mutual support. The IP is worked out ahead of time by the university and Genentech. And by setting that road up nicely, then what we're able to do is get real feedback, because one of the things that stood out to me in that collaboration was we made the molecules, they looked pretty good, but in the early version, and our collaborators said, yeah, but that looks really strained, like super strained. In practice, we've tried a whole bunch of public techniques, and the molecule, it has an energy that's just completely unworkable in practice. And so it's having at least a champion on each side and people working together directly would be my answer to you on that.
No, I can just say that we are in France. We are promoting a lot this PhD program where you have industry and academia working together. So we are really try to teach students to embrace industry and think about all the positive things you can have, like access to clusters, access to money, to experiments that we may not have in academia.
One more thing I would say is that a lot of the startups that are trying to bring innovation in the field of AI are often spin-offs actually of academic institutions, and so that in industry academia collaboration is almost inherent to a lot of the innovation that's ongoing. Now your question was more towards pharma and biotech at a large scale. I think that pharma and biotech can be a difficult industry to penetrate initially when you're a startup. They're looking for usually a lot of validation, a lot of very early data. However, I think in the field of drug discovery, in particular AI applied to drug discovery, you see a lot of different partnerships that are actually ongoing. AstraZeneca with Benevolent AI, for instance, is an example. Okin with Sanofi is another one. There are a number of partnerships of that nature. So I think we're going in that direction more and more, and pharma companies realize that they can embed AI models and products very early in their development.
So you think that in 5 years from now, there will be more and more drugs that are in phase 2, phase 3, that have been like discovered thanks to AI?
Definitely, yeah. This is already happening. They're already in the pipeline, but very early. So we'll have to. Time will tell.
Yeah, thank you.
Thank you. I'm happy because just a quick comment before. There is a lot of ethical consideration, and I will be hosting the ethics and AI next week, and I see, like it's very tomorrow, and it's very relevant. But anyway, my question was more as a cell biologist. And we generate more and more like big data sets, like omics data and all of that. And we build model and test those models. But kind of on the other flip side, where you were mentioning, like, usually we want like an end product and an end prediction with AI, but can we flip things around a bit and use AI to help identify gap in our knowledge? And how should we build framework to help actually scientists trying to make sense of those very large data sets that are very complicated and have a lot of connection, and help hypothesis generation, if you have any opinion on that?
All right, I think there's two levels of answer. First answer, the kind of glib one, but one that we want to get to working, is all of the explainable AI techniques. Interpretable methods, saliency mapping, occlusion, or even building models that operate through rationale layers, where you're having it solve multiple tasks as a series of steps that are fully differentiable all the way through. And if the model works or does not work through that information gate, then that tells you something, because you can change what information the task has to pass through. And so that's one category of answer, a technological answer, an architecture style answer. In practice, much of the saliency mapping methods, integrated gradients, guided GrabCam, either bring in their own artifacts or smear out signal, and can only tell you really basic things, like, is it looking at the dog or the background? But trying to understand about the ear or the nose is iffy at that resolution. And in molecules, goodness gracious me, we don't even have as much intuition. And so that answer is where the field is trying to go methods-wise. The flip side answer is a simple one, and it's a logical answer instead, which is, this is just one technique. And if anything, use very simple models wherever you can. Stand it up, swap in and out statistical models and other models, and ask whether the bang for the buck is worth it to bring in something that we have less of an understanding of. And so that goes back to being scientists, and the fact that all of us here in the room have agency in making that type of decision without having to rely on hoping that a model that learns for reasons we don't know can tell us something new that we didn't know to look for. So it's considering it as parts and swapping the parts.
No, I would just say that maybe one way to look at it is to use these models to validate an intuition that you may have. So the question will be clear. It will not identify gaps, but it will confirm or infirm an intuition that you may have.
One last thing. We mentioned earlier in the discussion the difference between supervised approaches and unsupervised approaches. I think that plays a role in your question. When it comes to identifying gaps, taking an unsupervised approach to look for high-dimensional structure, global and local structure in high-dimensional space, using what is, in many cases, now known as foundation models, so models that are looking very generally at a field, and then can be trained more specifically for a given application, is an approach that's very relevant. You can really, that's what I would say, just from the ML training approach, just being unsupervised or self-supervised, helps a lot.
Thank you. I like that you said intuition. It means that likely scientists are not going to be replaced by AI anytime soon.
Hi. So my little thesis was in proteomics and omics data at Stockholm University. I wonder one thing. How can you deal with the biophysical properties of a shape-shifter proteins like spike? Because we had a lot of problem to read the cryo-EM data. So I'm talking of a particular class of proteins that they are able to change conformation because of pH.
The short answer is no one's dealing with that yet. The ways you could go about trying to start would be to think about your representation. And because when we start a model, in order to do any deep learning or machine learning training, we have to make a decision first. And this gets to MLTwist, for instance, and thinking about what our representation of our information is. Not just the file format, but what is this cloud of atoms? What is this as far as the model is going to ingest it by the time it sees it for the first time in the world? And so geometric neural networks might be good. You can make graphs, spatial graphs in space. And then who's to say that a graph only has to operate in space? It can also go across time. You can put edges together to have ensembles. So that's something similar to that convolutional approach, but in multiple dimensions, and now you're convolving in non-Cartesian space. So that would be one way to do it. You can use attention as well. So there's a lot of methodological answers. Oh, but
Yeah, I want just to tell you that for desperation, we started to look at possible binding sites for transcription factors.
oh yeah. Well, let's continue this after, because
that's
there's a
fine.
bunch of
Thank
neat
you.
methods that can be done with this.
Thank you. No, merci beaucoup.
Good afternoon. Wonderful discussion by the panel. My name is Bhushan. I am a bioengineer and I develop biomaterials and cell therapy. I'm new to AI in bioengineering, so pardon my naive question. So I have a two-part question. One is, what's the panel's take on model hallucination and how it impacts the clinical data or clinical output or in output in cell biology as well? And the second part of my question is, we are talking more about going higher dimensional in terms of analyzing object or making new objects by the means of generative AI. But in terms of clinical translation, since I work on a translational side of a science, we want to have a simplistic system to be clinically translatable. So what is the panel's take on dimensionality reduction going forward after we have a lot of higher dimensional data set?
Based on the nodding. The first part of your question, sorry.
Model hallucination.
Yes. My experience with it, my humble experience with it, is related to generative AI specifically, right? I think that model hallucination is not really a concept that was really used until really that spiked a lot with stable diffusion models and LLMs. So when it comes to model hallucination, I think what we want to be wary of, or what we want to have in mind, back to what we said a bit earlier, is explainability on the one hand and reproducibility on the other. So being able, false discoveries is inherent to science, right? There are always false discoveries. The fact that other scientists, other researchers around the world can reproduce the experiments, reproduce the methodology, and find new results is really what corrects the course. And making sure that we push as an industry and as an academic community for the availability, again, of the models themselves, their code, but also the availability of data sets to reproduce and create new data sets in the future, I think would help alleviate a lot this concern. So that's really my take on it. When it comes to dimensionality reduction, I'm not sure I followed completely your question, but depending on the use case, depending on what you try to optimize, especially between the local and the global structure that you want to preserve when visualizing the data, there are different methods that you may want to use. In single-cell RNA-seq, QMAP is really something that is used a lot. In other fields, other types of dimensionality reductions are used. So my experience with it as well is really to focus on what do you want to retain as a priority from the latent space and really optimize for that, and trying different methods if needed.
So in the area of dimensionality reduction, one of the answers always used to be PCA or VAE, variational autoencoder, where you try to compress information and then reconstruct it. And conceptually, in both cases, you're asking what the variance of your data is and having a good representation of that variance so you can build it back in the autoencoder case. There have been interesting advances recently that might help in this space where, if I'm understanding correctly, your concern is it's a very large, sparse, and diverse feature space. Because if you're thinking about medical records, you're thinking about individuals, we have a lot of different different information about each individual. And that's a problem with the classic issue in machine learning where you need to have the same features, the same inputs for everybody. And so there were early versions, there's a thing called Deep Patient 7 years ago, maybe, that it was a VAE for patients where you just have a fair amount of noise and you do it's called a denoising autoencoder, where you inject random attack on the information and you ask the model to fill in the gaps. In the modern era, we call that masking or self-supervised learning. And so you can do self-supervised learning in an autoencoder context to help reduce your data and have a stable representation for a lot of very different patients that have a lot of very different information. You might also think about, and this came up actually yesterday for those who were here, this idea of contrastive learning, which is an example where you look at pairs of information about the same patient, or you take pairs of information about different patients that had the same condition, if that's what you care about. And you can begin to, in sort of a self-referential way, build up a better condensed representation, also known as embedding or a latent space, to do that. So that would be one way to think about dimensionality reduction. In the case of hallucination, absolutely. I mean, hallucination is generative. Otherwise, it's just a bad prediction. And kind of the answer to both of those is, we're scientists. You know, we go test it. And so if you believe a model is reliable enough to go test the predictions, it's an empirical answer to how we think about hallucination. And based on what we see, you can do active learning feedback loops in order to improve the model where it makes the most errors.
Yeah, thank you. Thanks.
Hi. My question is also in association with the former two questions which were asked about the difficult problems in biology, right? So it looks like once LLMs came in, everybody took out like the simpler ones, and they're also important because we haven't really solved like docking-based predictions and stuff like that. But what is the next frontier of biology beyond that, and what is stopping us from going there? Is it something at the level of like multiple models talking to each other, or is it simply the compute power? And if you could talk to like building realistically, teaching a machine biochemistry, where are we from there? Yeah.
Okay. So, in the one hand, there's the relatively simple question of, hey, when are we going to be able to actually make models that can predict if this will be a good drug, even if we already know what it is and we already have it in the pocket, and even then? And the issue is training data. It has just been terrible, and we're so limited of having enough data to train models at this scale. Even for really simple things, say I want to train a neural network to recapitulate something we know how to do because it's an existing docking program that doesn't give us the right answer, but at least there's a formula. In what we've seen is most state-of-the-art models need 80,000 or more training examples to recapitulate a simple sum of log energy style docking function, which is for sure way simpler than reality is. So we need way more than that, orders of magnitude more than 80,000 training examples. We're not yet at a place where it feels like we can get that from experimental binding affinity. And so maybe an intermediate solution would be some of these things called ABFEP, absolute binding for energy perturbation calculations. Problem is, you can only do a few of those calculations per day right now using classical methods. And so there's this problem then of, we really want this as a way to speed things up. We want to make sure we can do it in a generalizable way. We don't have enough data. The other, so that is kind of where we're at, and I think we just need a huge amount more labels in that space, realistically. There's ways we can cheat and try to get ourselves to kind of build up a foundation using these techniques we talked about that are foundation model-based, that are unsupervised, that are self-supervised, to maybe solve some of the basics of geometry first, and then have the learning process really focus on this really hard part of the question. But there's another sideways answer I want to give to your whole question, because I just answered a lot about binding affinities specifically, which you mentioned at the end, but wasn't the whole question. I would actually say the biggest advances we're about to see is the final mile problem. So many things are possible right now. So many models are sitting on GitHub, as we talked about and are shared, and so few people are able to use them. Or it's the same people every time. It's the same community. It's the computational people who made the models, who honestly made models that weren't solving the questions people care about more often than not, unless there's that collaboration, that relationship. And so that's the final mile, like in postal delivery. It's really easy to get it to your zip code. All the money and difficulty of a postal service is getting it to your door. So what is it necessary to get it to the bench? And so my best answer is this people collaboration question, but if we can close some of that gap, and I know that's a space that ML Twist is in, and also in the application and use of it, I think that's where you're going to see the biggest change. If no single new model will develop today, there's so many uses of the models we have right now that nobody is doing yet because of this friction.
That's enlightening.
Very briefly. I think infrastructure actually is critical. We do, so everything that you just mentioned is brilliant. I think what another frontier that we should look forward to is integrating the different modalities of biology right now. We do have very different lenses that we can use to look at biology at the DNA level, at the RNA level, at the spatial transcriptomics level, the proteomic level, et cetera, and putting all of them together, creating really a model that, a mental model and a biological mechanistic model that can put all of these layers together is really the next frontier. That requires data curation and data collection to a level that is extremely challenging, and that requires infrastructure that is extremely difficult also to scale and to put together. So that's really also something to look forward to, is the creation of these databases at scale and these intuitive tools for people to be able to access the data without having to be computational people only, right? We need the biologists, we need the non-AI and tech people to actually look at that data.
Well, thank you everyone for those great discussions. Thank you. Now I invite you to take a break, and then we'll gather again at 3 for the second panel about AI and drug discovery. Thank you.
Let's do something. Just call them up and say, hey,
raising the question.
Yeah, of course, that's a lot of money. Yes, we don't have that. We are now fully digital.
The world is never going to sleep like that.
Very recent. And in terms of basis, now massive funding for building.
Now they can see it better.
Just give me a second.
Yes, send me an email.
Hello, Katie. Julien. Nice to see you in person. How are you? It's like one weekend. How are you? Oh, so you don't have it over there? I mean, sorry, the department.
Well, all of my faculty are here. I would say most of my faculty are here.
And it's like there's no space in between.
Since COVID,
no one else working virtually. Yeah, maybe I'll try that, but now you know. You then also some.
So I've been working since 17, almost 15 years. So it's very a small crew. So we work as well. So it's very unique. You know, we never build a large corporation. We build like biotech company that will be designed to work with 7 people. So we don't
scan, whole body.
But otherwise, I do.
You do. They should be fast. I said my daughter's a writer.
Here at UCSF,
we have by 5 years. Her job is to sit quiet and write.
I guess that makes sense also, but I think that this was open on for students to be the career.
That
for,
is
for us, when you go around,
you're
even as a method,
you're right.
how do you do it?
No, many things got here, and they don't see a faculty member. And that's not true.
How did that bring you
Yeah. So, we've been sitting on this for a while.
For so long.
Wow, excellent. Yeah, well, of course.
And I love that. So then, I got through college for, I was ready for an encore career, retire, do something, at some point. But, so this was challenging, trying to do new things with pharmacy. But I found that, when I'm an academic, there's an idea, people rise around, you get funded, and you go, here, you think of something, I'm kind of bringing everyone along.
That's a different job.
You have to meet with people, and you have to make sure they buy into it. You have to, it's a whole lot to just move things forward.
You're in the spot, everybody.
Yeah, that's true too. I get all this too. Now it's muscles, Israel, just sending
Oh, you
them
see
out.
like
Oh, they send me that. Oh, I got that. I got like 30 emails.
what do you do with that? It's even your function to do
Yeah,
it.
and not only that, what am I supposed to, they're telling me what to say. Well, you're going to tell me what to say.
Oh, yeah,
So I'm a little
the
bit
PR
like that.
people coming in.
You're going to tell me this and that. I'm not going to do that. I'm not a politician. So I advise on campus what to do, but I feel like the most important thing is to make everyone feel safe and to make them feel like they can express their views and to make sure that's safe.
It's funny because that's really like American campuses. version in France, we don't have that.
You don't?
No, not yet.
And they don't want to in Express? Or?
That's not like that.
Yeah.
In France, we only go to the conference very much. I mean, meaning like here, the life,
Because they're in the dorms.
yes. In France, you actually, it's not what you're going to do in town, so it's not,
You
it's
can
not like
get
a
an
small city.
apartment. Yeah,
So it's not
exactly.
the same dynamic. So it's really, we look at this, that's really happening.
Well, here he is. I recognize this guy.
Hello. Ron, Julian, nice to meet you.
Happy.
Good to see you again.
Nice to meet you. Okay, we're going to have fun. Yeah. And our whole goal today is to be entertainers. Yeah, they're all That's my fault. Sorry. Let me start. There. Yeah, I won't. Don't worry. I mean, what I say, you can say.
Okay.
Right? Because sometimes I'll turn, if I'm here, I'll be turning my head to the right. That's a little to the right. Yeah. I sort of made the mistake.
Oh, you can come here.
Yeah.
I ended up taking it over because frequently in the middle of the day.
You have
that close to this.
I am here.
I had to bike to the station. And it was raining pretty hard.
Okay, here's Jamie.
Alright, I don't think we've ever actually met in person.
Not by phone, right?
But yeah, so you're on several
Zoom,
times.
so
Yeah. Hi.
Nice to meet you, I'm Julian.
Check, check.
Hi, Julian. Nice to meet you. Yeah.
Yes, ours all stayed in the massage.
Yeah.
If you want to say, happy to do a bad job, let them take those questions. Please do. We're just going to sit here, and then we'll pass those by.
I thought I saw someone else, so I wouldn't have been devoted to such a
Yes, we did that. Hi, I'm Celine.
Good.
Bonjour, Céline.
We're virtual. I have affiliations with some companies for the Parvus. I thought I was meeting with Lorena yesterday, but so this was Jennifer Williams, current contributor.
This is separate.
I don't see, can I put my stuff down?
Yeah, I put mine somewhere.
I just put it in my I didn't know you were too. That's all her fault.
We've lost a lot in rapid succession. Teja went to Brown. Deanna Kurtz was chair for 9 months. How long was Deanna chair for? Less than a year. What? And then she went to Ohio.
So, what's your rates?
Yeah, so there is a
Fast.
It is.
Oh,
Yeah, together. Oh, yeah, so you get together. Like caviar.
what did I get?
Okay.
Which is that picture was taken like 13 years ago. She had the last. There's a small budget. It's 5 years.
Yeah.
The big deal.
Yeah.
Sort of like yeah, and I don't think it's after 5 years.
No. Oh, no. I mean, this whole town, I moved here in the original cohort that moved. It was like we're in a field. I'm over in Rock Hall.
From the highway, you
Yeah,
come to it.
exactly. And there was a golf thing there, and it was just, it was very weird. Yeah, no, it's, it did change. It's really nice. I mean, I had ambivalent feelings about the basketball at Chase Center. I felt like it was a biotech. Didn't win that one, but still, it's okay. You know, that gives me a lively environment. I bring people over, we go to have fun. Yeah, that's good. But what skills do you have? Like you're like a chief executive officer.
But by training, I'm a PhD.
So you're a PhD, not an MD.
No. I learned
Do you wish you were?
no, not necessarily. In my experience, I'm compelled.
Okay.
On that side, the corporate side, yeah, for today, I'm just like the PhD side of the work.
And the drug development part, your first company, was it a traditional small molecule company?
DNA molecule,
Okay.
now.
Oh,
that's
something.
Yeah.
How did that work?
Well, that was well, we demonstrated, we were so, we did develop a platform in this company, and we demonstrated to clinical trial.
And it's not approved or is it?
No, so basically, yes, it's been sold to another laboratory.
Okay.
And then I'm done Christian.
Okay.
And where the asset is today, I don't know, because the service company is
Yeah, so you don't know.
but it was fascinating actually.
Yeah, I'm going to ask you those questions about, you know, how AI can help accelerate or expedite that kind of process. Yeah. 15, 20 minutes at the end. So we'll have our questions, and if we run out, fine. If we don't run out, we're going to stop. Okay, got it. Good. I think we're just going to sit down.
Yeah, so maybe I can take one share maybe?
Yeah, take one out.
Well, I think there are 4 of us. Yeah, if you're sitting here when you...
That's it. Oh, yeah, I don't want that. I don't want that.
Oh, well, next to you.
You can kick me under the table.
Okay, thank you everyone for coming to the second panel of the day. So this panel will explore the impact of AI in drug discovery and will be moderated by Katy Giacomini, the Dean of School of Pharmacy here at UCSF.
Okay, thank you. All right, so thank everybody here for listening to our panel, which hopefully will be fun and exciting, and will move AI into the direction of drug discovery and development. We have 3 panelists. We have, and I'm going to let them introduce themselves. But we have Jamie Frazier. We have, I'm going to get all your names wrong. We've got Ron Dror, and we have Julian Mamet. And so let's start. Just give a brief introduction on yourself, because then we have a lot of questions. Jamie.
Yeah, so hi, I'm Jamie Fraser. I'm a professor here at UCSF in the Department of Bioengineering and Therapeutic Sciences. My lab is on the 4th floor, directly above here, and we use a lot of the structural biology facilities, NMR, EM, crystallography, that are located sort of behind the atrium here. And I'm a structural biologist, biophysicist.
Okay, great. Thank you.
I'm Ron Dror. I'm a professor at Stanford. My primary appointment is in the computer science department. I have courtesy appointments in the structural biology department and the molecular and cellular physiology department. My work is computational, relates to structural biology. We use various methods to study structural biology, including physics-based simulations and artificial intelligence methods. And I will say up front that although I'm a computationalist, I believe very strongly in collaboration with experimentalists. Essentially all my work is in collaboration with experimentalists, and a number of those experimentalists are here at UCSF.
Thank you, Ron. Hi, everyone. I'm Julian Mamet. I'm the CEO and co-founder of Core Biotherapeutics. I'm one of the French in the room here today. By way of training, PhD in molecular biology and genetics, and 20 years of experience in drug discovery, drug development. So today, that's perspective I can give. I'm not an AI expert or a programmer by any means, but direct experience in bringing drugs to the clinic and through the clinic. So very happy to be here today.
Okay, great. Thank you all. I'm going to start with you, Jamie. And I guess the first question I'd like to ask is, you're doing a lot of work, and you've been studying, for example, protein-protein interactions. How can the studies in your lab and the research in your lab actually accelerate or enhance or help the drug discovery and development process?
Yeah, so I guess I'll break down that, and how is AI helping our lab accelerate the early stage drug discovery process. So the first way has really been in all the explosion of methods around prediction of protein, macromolecular structure, and really the confidence boost that we have now with AlphaFold-like methods relative to the situation a few years ago. This has made all the boring parts of structural biology go from taking weeks to taking seconds, and has really accelerated and allowed us to really focus in on, I think, the more interesting parts. That's in more challenging complexes, but also in higher throughput, makes it much faster. The other area that I'm particularly interested in and using AI for, and that I've been particularly influenced by, I think, our mutual collaborator, Brian Scheuket here, is in thinking about how AI can help us explore chemical space more rapidly and more completely. And here, I'm really interested in this problem of thinking about a target. There is a molecule that can be made somewhere in the universe that would be the perfect inhibitor of that molecule, that would also have great PK, that would also not hit any of the anti-targets. It's just up to us to discover it. And the trouble is we're very inefficient at searching that space. So we're really interested in active learning methods to do that. And something that I've really been impressed with Ron's recent work, thinking about how generative AI methods can start to crack that problem, which is, I think, the next frontier.
Okay, thank you. Ron, do you want to comment on Jamie's comments there? And yes.
So I agree with everything Jamie said. Well.
Deny everything.
No. Actually, no, I really do agree with everything Jamie said. I will try to add a little bit. Basically, one thing that's been a focus of mine for a long time, for the past 20 years more or less, is that having a structure of a protein or other biomolecule is a wonderful starting point. Structures are very informative. When I say structure, an experimentally determined structure or a computationally predicted structure. But one wants much more than that. Proteins and other biomolecules are highly dynamic. They change shape over time. They're constantly wiggling around, and those dynamics are really important to their function. As Jamie said, they're constantly interacting with other molecules. That is, proteins interact with other proteins, with RNA and DNA, and of course with drugs. Those interactions are really important. Structures provide super useful starting points for understanding all of these things, for understanding how proteins and RNA molecules work, for example, how they interact with other molecules, what drugs do to them. But just a single structure doesn't tell the whole story. So much of what my lab does is going from a structure to a more complete picture of what these molecules do.
Okay, thank you, Ron. Okay, now I'm going to turn to Julian, who I know is working on more complex data sets. So can you talk about how AI will contribute to analyzing those complex, or how you believe AI could contribute to analyzing those complex data sets, and get us more to precision medicine as well?
Sure. So maybe starting with precision medicine and then backing into what could be done here. So precision medicine is really kind of aiming at identifying certain form of disease biology that subgroup of patients have and developing treatment for those particular patients. Historically, that was done through, and you need biomarkers to do this. So historically, that was done with genetic markers. One mutation was identified to drive really a disease that allows to develop drugs for this particular biology. That's pretty successful. One of the actually very recent success in that comes from a friend of mine at Syndax, which is a mid-sized biotech company out of Boston. They just got 2 phase 3 approvals for a menin inhibitor for treatment of a subpopulation of AML patients that have 1 of 2 mutations. And it only works in this 1 of 2 mutations, basically. It represents 10 or 15% of patients. So that's one of the cases. If you push precision medicine to an extreme, now the FDA is promoting developing not precision medicine anymore, but individualized medicine. Develop 1 drug for 1 patient. And they promote that through oligonucleotides that could address a very well-characterized mutation that drives the disease. So that's actually been done already. There's been an antisense that was developed for a young girl that had a neurodegenerative disease, Batten disease. Her name was Myla, and they developed an antisense to treat her. It's called Mylasen. So that's precision medicine. That works well when there is 1 cause to the disease or 2 causes to the disease. But most of the diseases are multifactorial. So how do you identify biomarker that reflects a particular biology? So that's where AI can come in to do what it does right now. A lot of groups are doing, trying to do molecular profiling of patients. And there's a lot of companies doing that, like Curie Life Science. It's one of them leading that space, having hundreds of thousands of just omics data, from DNA sequencing, single-cell RNA sequencing, and the like and the like. And they're trying to extract new biomarkers, new profiling for these patients. So AI really enables to bring precision medicine beyond like the 1 or 2 markers.
So it's handling something more complicated than
Oh
we
yeah,
used to
absolutely.
think of.
That's sorting and organizing large amount of data, basically. Yeah.
Okay, so all of you are bringing some unique expertise here, and Jamie, you mentioned collaborations at the very beginning, and also Ron mentioned collaborations. And so, Jamie, can you talk about if you feel that there are, how collaborative efforts between all scientists can actually, all boats rise, everything gets better, especially in the drug discovery area?
Well, yeah, I think especially as relates to incorporation of AI methods, I think actually the hard part for those of us who sit a little bit more on the experimental side is thinking, how do we get our data and our sort of pipelines of generating data really ready for artificial intelligence, machine learning methods. And so that means thinking about generating data at scale very differently than we would before. That means a little bit less data just to prove one specific hypothesis, but rather to flesh out sort of a model's understanding of the data or of the system. And I think those are really shifts that I don't think most labs are really entirely equipped for yet, but that our new entering graduate students are being confronted by this reality where, hopefully in 3 or 4, by the time they're ending graduate school, they will have sort of, AI-driven experiments that are, frankly, a little unintuitive to us who are used to a more specific hypothesis mechanism-driven experimental mentality. And so that's really exciting, but it means that there's a big challenge for experimentalists to get ready for that and start to shift our mindset a little bit.
Right, how you think about it, how do you approach it? I mean, if there's a model of a human being and here's what's happening, it's easier to design experiments. Here it's going to be tough. Okay, okay, I'm going to ask each of you, I think all of you here, if you could just look at molecular biology, especially the molecular biology that's informing all this drug discovery, ask where AI, answer the question of where AI you feel has made the most impact, the biggest impact at this point. And should I allow AlphaFold to be one of the answers?
Sure, but
okay.
Maybe just to be a little, provocative before I answer that question, I think that's a little like asking, this is a building name, Genentech Hall. This is like asking in 1985, where are restriction enzymes going to have the biggest impact in molecular biology, or something like that. AI is now just part of science, and it's going to be, there aren't going to be we're not going to sort of refer to these specific things as much as thinking about how all of molecular biology needs to be AI ready and be interpreted in an AI first complex. So, obviously AlphaFold Multimer is the answer to your question.
Okay, Ron.
Yeah, I agree. It's hard to say. And actually, if you take something like AlphaFold, so I'm friends with the people who did AlphaFold. I think it's wonderful work. I think it will lead to a lot of other great work. But if you just take AlphaFold in its own right and ask, what's the impact on drug discovery? It's kind of hard to say. The fact is, it produces more structures, but we've had lots of experimentally determined structures for decades. And most of those are more reliable than AlphaFold models. And it's not like they've just led to floods of drugs. So I think of it a little bit more by analogy to, say, the Human Genome Project. The Human Genome Project was a major achievement. But when it was completed, it was sort of hyped as, this is the solution to drug discovery. And then there was a lot of disappointment because drugs didn't pour out. If we look now, then without a doubt, the Human Genome Project had a huge impact on biology and on drug discovery because there was lots more sequencing afterwards. It's just, the fact that we have all these sequences now makes a huge difference. But it's not just an immediate, here's the solution. So I feel like that's true for AlphaFold and all its competitors, all the other machine learning methods for structure prediction. But I feel like that's true for a lot of other things that are going on in AI now in the drug discovery space. They're not just going to kind of solve the problem right now, lead to outpouring of drugs. I see it more as very exciting, ongoing work that will have a long-term impact, but it's hard to say exactly where that impact will be greatest.
And maybe if I can just, even with AlphaFold, I would say the greatest impact on drug discovery that AlphaFold has had, in my observations, is in construct design. It's not in docking to, a specific small molecule to a predicted protein structure. It's to enable experiments to go faster. It's been like really mundane stuff of, we should trim back this disordered segment or cut off this loop or what have you. It's been in really mundane things that have very little relationship to the sort of grand ambitions that, DeepMind might write about. Right. And that's true of a lot of, enabling tools, is you don't even know what people will use them for, and, they'll accelerate a lot of mundane things, and that really matters. Those cumulative advantages really add up.
Okay, let's let.
So just to be totally clear, I think AlphaFold is wonderful work. I think other Rosetta Fold, etc., like all these, it's just really wonderful work. But I think there's a little bit of an expectation in the media that let's just solve the drug discovery problem, and that's just unrealistic. And for what it's worth, I don't think any of the scientists doing this work are making those claims. It's just other people like to tell the story that way.
That is true. Jill.
I'm glad you guys talked before me. I completely agree. You have to define impact when you're asking that question. And if you look at this with the whole drug discovery development prospect, the impact is, did you succeed? Did you went through the 15 years? Did you get phase 1 approved, 2, 3, whatever you had to do, and get your approval, like Syndax I was just mentioning? So the problem is, I used to say that if AI generates a drug or my cousin generates a drug, it doesn't matter. At that point, it's a drug that's going to go through the whole process, and then it's going to find a number of confounding factors through the whole thing, preclinically, clinically, that can mask its activity. So it's going to be extremely hard, extremely long to actually determine the impact of all of these. Ultimately, you're going to need, maybe dozens of drugs that are AI in their design or development that are more successful than others to start to change the whole system, if you want. Right now, the field is just too nascent. There's just a handful of drugs that have reached phase 1, phase 2, actually maybe 4 or 5 I have in mind. Half of them have failed already in phase 1. So it's no different at that stage than how we develop them typically. So it's just going to take a long time. So it probably helps a lot, very specific bottleneck, and answers the question of those bottlenecks. And that's where, when you're looking at this particular problem, that can enable a lot of things to be done. But then when you put it in the big picture, it's just, you can't really answer this question now. We have to come back in 30 years.
No, I'm hearing you. I always thought, this was just my thought, that that drugs failed quite a lot early on because of toxicity, unpredictable toxicity that may occur, and it may occur. Can AI help that problem?
I guess it all depends. That really depends on what you're doing. Almost every disease you're going to target has a different development path. If you take antibiotics, antibiotics, if it works in a petri dish, it's going to work in the human body. But it's all about toxicity. You're going to go into phase 1, you start to develop a little rash, you're done. That's it. You're not, but you're done. So, and that's how it works. Now you're developing drugs for cardiovascular disease. You're going to have to test 10 of thousands of patients. Yeah. So just the time it takes, take relaxin. Relaxin has been in development for maybe 40 years. So many companies trying to find indications. The last one was cardiovascular. And, friend of mine, just company get acquired by Novartis, and for relaxin, Novartis take it to phase 3 trials, failed. 10,000 patients each trial, they failed. So it doesn't fail just in phase 1. It heavily fails in phase 3 too. So
that's worst. Maybe. Okay. Something we all grew up with is garbage in, garbage out. So can you talk a little bit about the quality of data and how that affects this machine learning, and can it cause things to go off rails, and then comment a little bit on quantity of data. And I think this question is for anyone who wants it. So Ron, Ron, can you tell me about quality of data? And how can you talk about some model that went off rails because bad data was put in or something?
If it's okay, I'll talk first about quantity of data.
All right,
so I actually did work in, while I was a PhD student, much of my work was on machine learning. Machine learning is the subfield of AI that has led to pretty much all the recent advances. But back then, machine learning, it was a subfield of AI. It had been a subfield of AI for quite a while, but it hadn't advanced all that quickly. And if anything, at the time I felt like it was perhaps underhyped. Now I feel like AI is perhaps overhyped. One of the things that gets swept under the rug is that the great majority of the recent advances in AI, especially the ones you read about in the popular press, are enabled by huge quantities of data. That's actually one of the things that is perhaps the single biggest thing that made machine learning go from a niche area to something mainstream whose results you read about all the time. The fact that for text, for example, one can now get nearly unlimited amounts of text very cheaply from the internet, and that's a big part of what enables something like ChatGPT. For image recognition, one can get, again, nearly unlimited of quantities of text, of images very, very cheaply from the internet. And with digital cameras, you can now collect more if you wish, very cheaply. This is true in many different areas. But one thing to keep in mind is that there are many problems we want to solve where there isn't a huge amount of data available. I actually think that's true for many aspects of drug discovery. That doesn't mean that artificial intelligence for those problems is hopeless. After all, humans are actually really good at making good inferences from small amounts of data. But it does make the problem a lot harder. Sure. So bottom line is a lot of current AI approaches are predicated on the assumption that we have huge amounts of data to work with. And I think we actually have some really hard work to do to make these approaches work for small amounts of data. And that's even if the data is of good quality.
Thank you. Anyone else want to add to I think what you're saying, or what I'm hearing you say, is that quantity quantity itself can almost overcome quality, right? Right?
Let me rephrase it a little bit.
Okay.
I would say that most of what's currently being done in AI relies on large quantity.
Yes.
And so the challenge is to make it work with smaller
Something
quantities.
small.
In terms of quantity versus quality, I don't think it's the case that quantity can overcome quality. What probably is the case, at least for some applications, is that if you have a large amount of low quality data and a smaller amount of high quality data, then you can make things work.
Okay.
If you don't have any high quality data, there might be a way to make it work, but I don't think we know it now.
Okay.
I think the garbage in, garbage out
Right.
does apply. There are definitely plenty of examples of AI researchers taking, basically taking poor quality data and making poor predictions.
Yes.
And in those cases, honestly, it's hard to tell if the poor predictions are because the AI didn't work so well or just because the data was poor. But you definitely need some high quality data to work with.
Okay. Thank you. Anyone want to add? Okay, I'm going to move to the next question. You do want to talk, Jamie? No, don't let him.
There's another element to this, which is not just the quantity of the data, but also the diversity of the data. And so you were talking about sort of talks and AdMet problems before, and it's actually relatively routine to in a campaign or in a drug discovery project to train very good models that work very well in a region of chemical space that is of interest to that project. But then those models, do not generalize to the next target, right? Or even to an unrelated series hitting the same target. And, one of the real sort of magic moments for many of us using, ChatGPT or something like that is this sort of feeling that there is generalization going on, that it's not just sort of learning very local rules or memorizing things, but that there's generalization going on. And so for us in the life sciences, we tend to have, we tend to be very good at at getting high amounts of high quality data in a very narrow region. And one of the things that we really need to work on are sort of being able to keep scale and quality of data while also being much more diverse in what we sample.
Thank you. All right, I'm switching back to sort of molecular biology, and I would like to talk about target identification and target validation. So there are many ways to discover targets. People do it through genetic studies, they do a knockout mouse, they may do a lot of things to discover targets. So can you tell me how AI, and this is for you, Ron, can you tell me how AI-driven approaches can enhance that? Target
I think Julian may actually be better.
Okay, Julian, let's go. Julian, yeah. And challenges also.
Yeah, many ways to identify targets. I can tell you how we do it, the approach, and it doesn't involve AI right now. And the reason is actually what you were describing. There's not enough data to build the science behind our platform, and through AI. We're at the point where we're considering putting it in, but we've been using network science and information theory. So we work on networks of transcription factors. One of the lessons we had through my previous company was that you can block a transcription factor efficiently in the clinic and get a profound effect, but it's going to be limited to a small amount of patients. So to really have broad effect across population of patients with transcription factors, we need to work with them, which is as networks. So we've been interested in answering a simple question, which is what is the functional dominance of every single transcription factors in a network of 1,000 transcription factors. So we developed tools to do that. Non-AI tools, but network science tools that we validated experimentally. So that's kind of how we do it. Just you start with your question and take the best tool. Now, the AI version of this kind of approach is people are looking at omics data, just again, next-gen sequencing, GWAS, single-cell RNA-seq, and the like, and they try to use AI to find patterns that you couldn't find as a human being. So that's one way of doing it.
Yes.
We are using AI. So.
Okay. Yeah, I find that interesting because we can only absorb sort of maybe the single experiment, the single GWAS, but this AI could put it all together and maybe find issues.
And look for patterns. I mean, just imagine just an Excel spreadsheet with just 100,000 on each side. You can't do it.
Okay. We're going to ask like one more question, and I'm going to open it up to the audience after that. So, okay, let's see. I get to pick one here. So I'm going to choose, what are the challenges in translating AI-discovered drug candidates from the laboratory to clinical trials? So now we are, I think, talking about the ADMET space. You've got it in the laboratory. Now you've got to go all the way to the clinical trial.
So translating a drug to the clinic, that means answering a specific set of, or trying to understand a specific set of parameters. One is your dose level. It's going to be your minimum efficacy dose, your maximum efficacy dose. Dosing regimen, how frequently you're going to dose. It's also trying to understand which patient population. That's one of the hardest ones. Where I go back to biomarkers. Sometimes it's just not available at all. So how are you going to go? Are you going to search through that? So it's answering this kind of very specific set of, and toxicity and efficacy. You would think that testing efficacy is trivial. It's not. In tumor, for example, or oncology, there are some objective measurements. You can look at a complete pathological response of a tumor. If you move into neurodegeneration or pain or subject reported outcome, completely different space. If you do it in pain, for example, are you going to ask a patient to rate his pain on a visual scale, on a numerical scale, do that at home, report it to a physician? All of these are confounding factors. So it gets extremely, earlier point, you get an AI drug that could be a beautiful drug, and then you're going to go into a space that has nothing to do with your drug, but with a lot of confounding factors you need to punch through. So what can AI do there? What I see is just companies developing now predictive models of trials outcome. A number of them, there's some in France, there's some in the US, there's some in Asia. And now they just use AI to integrate a high number of factors that, integrating the data they have about the molecule itself, the targets, the biology, what we know biology of the disease, and operational factors as well. How many clinical sites are in, how long it takes to enroll patients, all of these things that be confounding. So that's the kind of things that AI can do when you move away from the beginning, which is targeted identification, molecule development. When you start with the clinic, That's the kind of thing that AI is trying to help. So there's a lot of progress there. The FDA is actually pushing for this. So I do hope that sooner than later they'll be able to help us. Just to give you a frame of reference, in my prior company, to answer this simple question I was just going through, 7 years basically, and 4 clinical trials. So, and not always positive trials. We had to go through negative trials before getting positive trials, which creates a lot of issue, of course, when you're a company. So that's when you think back to the initial question, using AI is incredible, but it's going to take time to demonstrate the values through the continuum of the process. So.
Okay, thank you. All right, I am now opening it up to the audience for questions. Got some smart guys here.
If you have a question, you can line up here at the microphone. That would be great.
Hi, I'm Simona Zompi. I've been working in cell therapy, drug discovery, most recently at Sangamo Therapeutics, and I worked on iPSC. So one of the things that, listening to Julien about the transcription factor networks, how we can apply that to cell fate decision and differentiation, that sounds really exciting. And then the second question is more around, drug discovery and how we can make it better. And one of the place where it fails is, the predictability of the models we have, mostly in vivo models, are not very great, especially for cell therapy, where we have to build this humanized mouse model, etc. So how can we think about AI, using AI to make those models more predictable, and what do we have to change in our experimentation to feed AI there?
So the in vivo models, we have to live with them. Everybody hates them, but you just can't go, except very specific cases. You can't go to the clinic without them for many reasons. So how can we prove them? What I'm seeing right now is people trying to use AI to have a more holistic assessment of symptoms in an animal, in a rat or in a mouse, to move away from, for example, what we do in pain, which is poking the rat with something to see if he has pain. So that's the kind of tools people are trying to develop for those models. But it's going to be long. It's going to be this one. It's going to be difficult. And what was the first question? Sorry.
Oh, the transcription factor network for cell fate.
So we actually work on that, and iPSC neurons for ALS, for example. So, but we apply our own prism of what we saw determining the functional dominance. But what's of a network? But what's fascinating is, and we were not the one who saw that initially. There was some paper published a long time ago showing that transcription factor networks share a common architecture across cell types and disease. But those architectures are operated by different transcription factors. So, and they did that at kind of the module level of a small TF. So we do that at a big level, at the level of the entire networks. So we just find network mechanism that seems to drive cell fate. So I'm happy to talk about that later, but that's definitely a kind of thing that we're doing it, but AI can help doing as well through different, there's many ways of looking at it, but AI can actually help also looking through. So.
Hi, so I'm really interested in how AI can improve the mundane and administrative aspects of clinical trials, right? So we know that insufficient patient recruitment is a problem. And we know that, for example, language barrier or literacy barrier is a barrier for enrollment. So can you speak about the AI applications just for these?
Of course. So now you're in digital health, right? So it's moving away from the discovery, but trying to smooth clinical trial process. For example, going back to pain, one of the issue we had was just how you make sure that a patient that is in pain actually takes its time to rate his pain, because you need his data for the trial, right? So there's a lot, and when he takes his drug. So there's probably a lot of, I would say, AI process, on the digital side of things that could probably help making sure that the patients actually comply, and that you do that in a manner that doesn't interfere with how he's recovering from his pain. The traduction is actually a very, very good point. I mean, you do have to, when you start to do trials in multiple countries, that's a big piece of it. How do you translate a given English question into French or in Italian, and just like in a way that means the same thing? There's people living off of that right now. That's their job. So can you have AI standardizing all of this? So I think that if there's tools, probably could be developed for standardizing all of these. But I'm not an expert in this, but definitely, like any form of administrative process or operational process, it probably can be smoothed out for sure.
Thank you.
Thank you.
Thanks for the inspiring discussions. I'm a postdoc at UCSF. So I'm really interested to perform some basic machine learning or AI tool on my research. So I have a general question that practically, if I got some data sets like a small amount of high quality, a potentially larger amount of the low quality data, how could I really like perform some basic AI analysis on it? And in future, where the generative AI could come in, like for the analyze these kind of data sets.
So one basic answer is that there are machine learning methods like random forests and support vector machines that have been around for a long time that actually work well with small amounts of data. I would start with those sorts of approaches. I would take the high quality data you have, see what kinds of predictions you could make. With those approaches, there are software libraries. Basically, there's plenty of software available that implements these methods. I would try that before doing anything sophisticated, as in before going to a machine learning researcher and saying, what's the latest and greatest technique you can throw at this?
Thanks.
Yeah, I think the real key thing is always defining the question you want to ask, right? And you've described the classic, like, I want to learn how to use this hammer, question. And maybe it's like you want to learn how to use large language models or something like that, So then go find a question that teaches you, that will allow you to use those tools. Don't try to force your data set into a framework that, could probably be handled with less overfitting by a simpler technique. Yeah.
Thanks.
Actually, if I could just add one thing to that. So as I mentioned before, I felt like when I was in grad school, machine learning was underhyped, but now the pendulum has perhaps swung in the other direction. I think machine learning and AI more broadly have tremendous promise. Of course, I'm biased because this is because I do that kind of work. But I think one thing to keep in mind is that there is a lot of hype, and the hype tends to, even within machine learning, the hype tends to go from one method to another. So right now, large language models are the thing. Before that, it was diffusion models, and at some point transformers, and at some point convolutional neural networks. And something I've noticed is that whatever the hottest technique is, there's a tendency among lots of people, ranging from students to far more senior people, to ask, can we apply this latest method to problem X? It's a reasonable question to ask, but I think it's very important to keep in mind that this latest and greatest method is not going to be the best method for every problem. Many times it will be a much older method, and honestly, in many cases, it will be a method that's not even AI. There are plenty of good computational approaches to problems that don't involve AI.
So I kind of have a twofold question. So first is, what do you think is the space of AI for drug repurposing discovery? And like, do we need also like to change our process of pharmacovigilance to be able to integrate that better into machine learning model to lead to better drug repurposing? And the other aspect that is also a bit linked to that is, can we also find ways to use AI with the previous question to identify like potential synergies between drugs for treatments?
No. Actually, back in the day, we actually partnered with a company in the Silicon Valley for that particular purpose, trying to help us find, drug to repurpose. But it's interesting because it's a different development path. I guess, more integrating phase 4 data or pharmacovigilance data, definitely the more data you have, the better it is. And I think, same thing, now you're entering a realm of a drug that's going to be, that's been out for a very long time. So there's a lot of data on that drug. So yeah, AI can definitely be used to just find patterns to identify new application for it. So that's just completely agree with the high level, and we tried it also. As far as the technical piece of that, I'm not the right person to answer. So.
And on the drug synergy aspect.
So the drug synergy, that pertains to biology now. That pertains to what are the targets, what is the possibility of those having a synergistic effect. Start with an additive effect. And just can you already have 1 + 1 = 2? That's already not bad. I mean, if you can give 3 or 4, that pertains to biology. So I guess that you're going to have to look at this through biology of the targets here. So yeah, but again, it goes back to the question you asked. Then you find the tools to answer those questions. There's no blanket way of answering that question. It's kind of case by case, depending on what you know. Are those 2 ion channels, channel transition factors, these kind of things. But yeah, that's typically the kind of, I guess, application you could look into.
Thank you. Hi, thanks for a wonderful discussion. My name is Bhushan. I'm in Department of Bioengineering and work in biomaterials and cell therapy. So my question is truly from the perspective of personalized medicine. Would like to know the perspective of a panel on truly personalized medicine, which means that one drug developed for one individual. So it may not be possible to follow through that drug through a traditional clinical trial pipeline. How you think the AI could help in developing those very personalized medicine, and where we are right now, and whether it is really realizable in the near future? Thank you.
The biggest problems I see in that is not the will to do that or the sort of ability to attack targets. It's the financial incentives. You see companies now pausing development on drugs that probably will work because the patient population is too small, because it's maybe thousands of patients a year. Never mind one. I guess we have an election coming up in November. Figure out how to get this these types of issues raised at a broader level, because the solutions aren't going to come from AI. They're going to come from a major rethink of the entire economic model of healthcare, not only in the United States but the entire world.
Can I ask a follow-up?
I completely agree with that. And in fact, without any AI, basically there are existing techniques like CRISPR that could be used to cure various genetic conditions. The biggest obstacle right now is many of these are rare, and for most companies, it's just hard to make it work financially.
Okay. No, I was only going to ask.
Maybe the optimistic note, right, is that maybe there are ways of grouping together patients, and things like that, that we're not seeing with our conventional analysis, that are sort of, again, generalized features that some different level of understanding will be able to get. And then maybe there's a path there for development, but then you've got a regulatory problem on top of that of justifying sort of that property. So it's like a fascinating question. I think we could spend the whole hour just on thinking through the consequences of your question.
Yeah. Thank you so much for that perspective.
Thank you.
Really appreciate it.
Okay, last question.
Let's go. Thank you so much. So my question is about metrics of success. So you said maybe it's a little bit overhyped AI. I guess the ultimate metric will be we get a target and moves all the way through clinical trials, and we have a successful drug candidate, and that will take quite a bit of time to see that through. So in the near term, I'd be interested in what you define as metrics of success now, maybe 6 months, a year, 2 years, and coming from academia and industry, maybe the differences between the two, or just your labs personally, what you think success for AI would mean for you.
What? The simple answer to this, from my perspective, is just, did it help you answer the question you had, just right in front of you at that particular stage? That's because it's going to be different every single stage. So really depends. You're saying that success is defined from succeeding in step 1, step 2. It's really a question that pertains to what you're doing. It's I don't think there's a broad answer to this. It's really case by case. But.
Yeah, I basically I agree with Julian. And in particular, in the popular press, there's a tendency to talk about AI-discovered drugs. The way I think about it is more that drug discovery is a complicated process that involves many different, there are many different aspects to drug discovery. There are many different things you need to do to, even to just come up with a candidate drug molecule, and many more you need to do to bring it to clinical trials, to see it through clinical trials, to make it successful. I think there are many different sub-problems where AI can help. And for each of these, there usually is some reasonable metric to use. If one poses a general question, is AI relevant to drug discovery? I would argue we could have said yes to that 40 years ago. Because 40 years ago, people were already using techniques like regression for predicting properties of molecules and for all sorts of other things. And in the master's level machine learning course at Stanford, the first thing we teach is linear regression. Because it forms essentially, of course, when people talk about modern machine learning, the techniques have become more sophisticated, but they're built on that foundation.
I also think academia has a really important role in this, which is actually we should care about the metrics of success in terms of running actual experiments. In drug discovery, no one cares how you got there. It's just that you got there, right? By any means necessary. Whereas, we are in a really wonderful position where we can conduct these experiments. We can order, 500 molecules designed by this approach and 500 molecules designed by this approach, or, put these things really to the test in a way where the goal is to generate knowledge and compare these things, not to reduce it to some practice that maybe doesn't even work for the right reason, and just get it to the next stage and the next stage and the next stage. So.
Okay, that was the last question. So, all right, so I would like to thank the panelists for just a fascinating discussion, and I look forward to more AI in the future. So let's give them a big hand. And thank the audience for being here. Thank you.
Yeah, so thank you so much for a wonderful discussion. And so we will come back tomorrow for 2 other panel discussions, one on generative AI in clinical practice and ethical consideration of AI in healthcare. It's tomorrow, same place at 9 a.m. And for those that are invited to the next event, we are around if you have any question. Thank you so much. This is you? This is Jack,
mais du coup, ils ont quoi? Ils ont un bus à 5 heures, c'est ça, à l'hôtel?
Gina, do you know about the bus?
It leaves from the hotel at 5.
Yeah.
Huh?
At the
From
hotel at
Luma at 5.
From the side. I don't know. It's a big bus. Or can we join?
Yeah, I think it's like yesterday, yes.
Yeah, because I don't have my car, so otherwise I would have went with my car.
Are you
but
sure
I-
it's
I don't
5?
have it.
It's not 5:50?
Double check with Gina or Simone.
What is the name? Luma.
Luma.
Luma. Yeah, I know where we had the drink once and stuff. Yeah.
And then tomorrow,
Yeah, especially with the simulation. The first session was interesting, but kind of overlapping a bit on ethics. But but anyway, it was interesting. On voit tout de suite le...
What do I do?
Yeah.